The title of today's lecture is Models for Anatomically Modern Homo sapiens, and most paleontologists agree that around 100,000 years ago, the world was populated by a morphologically heterogeneous collection of hominids. We had Neanderthals in Europe, we had archaic Homo sapiens in Asia, and we had anatomically modern Homo sapiens in both the Middle East and Africa. By 30,000 years ago, much of this diversity had disappeared, and the only hominid species left was anatomically modern Homo sapiens. The question is, how did this happen? What happened to the Neanderthals? What happened to the archaic Homo sapiens? Why did anatomically modern Homo sapiens survive versus the others? This is an example of how people believe some of the distribution of these hominids occurred. There are two primary explanatory hypotheses to account for the transition from multiple hominid species to only one. These are known as the replacement model and the multi-regional model. Just to give you a brief overview of each, and then I'll go into each one in more detail. The replacement model argues that hominid populations were genetically isolated from one another during the Middle Pleistocene. 200 to 100,000 years ago, anatomically modern Homo sapiens arose in Africa, spread out, and replaced all other hominids. This is the out of Africa model. Hominid populations, according to the multi-regional model, are linked by gene flow. Here, they're genetically isolated. Anatomically modern Homo sapiens evolved from different Homo erectus populations all over the world. The distances led to regional differences in morphology. So this is just a graphical illustration of the two hypotheses and how they are perceived to have occurred. So let's talk about the replacement out of Africa hypothesis to begin with. So the replacement model posits that there's little or no gene flow as anatomically modern Homo sapiens replaced local populations of Neanderthals, Archaics, and Erectus. The implications of this hypothesis are quite interesting. First, in Asia, it means Asian Homo erectus and archaic Homo sapiens in Asia have little or no genetic continuity with modern Asians. They were all outcompeted and replaced in Asia by anatomically modern Homo sapiens from Africa. This means that modern Asians are descendants of the first modern humans out of Africa. In Europe, what this means is that Neanderthals were replaced by anatomically modern Homo sapiens, and that modern-day Europeans, such as myself, are descendants of the first modern humans out of Africa. In Africa, similarly, Homo ergaster is the ancestor of modern-day Africans and all other anatomically modern Homo sapiens. And modern Africans are the descendants of anatomically modern Homo sapiens in Africa. Another very important component of the implication of this hypothesis is, when did human variation evolve? According to the out of Africa hypothesis, the variation that we see today between human populations, between human races, has occurred since the time of replacement. And remember, anatomically modern Homo sapiens occurred between 100 to 200,000 years ago. That was the time of replacement. So the variation that we see today in modern human population is relatively recent, with only averaging approximately 100,000 years. This hypothesis also states that all the genes necessary for the modern human phenotype were all assembled in Africa and spread out as people carrying the genes migrated out of Africa and replaced local populations. What about the other hypothesis? The other hypothesis is known as the multi-regional hypothesis. And the basis of the multi-regional hypothesis is that the genes creating the modern human phenotype arose in all parts of the world and were mingled together as people from different regions mated and exchanged genetic material. It argues that continuous gene flow, such that modern populations from different regions resemble archaic species from each region. So we're going to see that gene flow even just a little bit, is very important for the migration of, or the movement of genes from one population to another. What are some of the additional implications of this hypothesis? First, in Asia, Asian Homo erectus and archaics have genetic continuity with modern-day Asians. What does this mean? It means effectively that Homo erectus gave rise to modern-day Asians in Asia. In Europe, the implication is that Neanderthals and modern-day Europeans, such as myself, have genetic continuity, that modern-day Europeans are the descendants of Neanderthals. In Africa, it's argued Homo ergaster is the ancestor of modern-day Africans, but not other anatomically modern Homo sapiens. Additionally, in terms of the variation, the multi-regional model argues that the variability that we see today between human populations, human races, has been present since before anatomically modern Homo sapiens developed from Neanderthals, ergaster, and erectus. They argue that continuous gene flow means that the genus Homo never fully speciated. They were all able to interbreed with one another, leading to modern humans who were also able to interbreed with one another. So, most importantly, the genes for modern human phenotype arose in all parts of the world and mingled together as people from different regions mated, exchanging genetic material. Okay, so what kind of evidence do we have for this? Well, let's start out looking at the fossil evidence, and then we'll look at the genetic evidence. The fossil evidence for the replacement out of Africa model is that basically the earliest anatomically modern Homo sapien fossils have been found at both Kafka and School and in Israel, and they are dated to about 90,000 years ago. So we see anatomically modern Homo sapiens and Neanderthal fossils from the same time period in Israel. We also note that Neanderthal fossils have been found at Tubun, Kabara, and Amud, also in Israel, and they are dated to 110 to 60,000 years. This indicates that there's an overlap of at least 30,000 years for anatomically modern Homo sapiens and Neanderthals. It's been suggested that this 30,000 years is the amount of time it took for anatomically modern Homo sapiens to outcompete the Neanderthals and actually replace them. So that is inconsistent. The fact, let me reiterate, the fact that there is this overlap of 30,000 years is inconsistent with the multi-regional model, because the multi-regional model says that one gave rise to the other. The fact that they occurred at the same time is support for the out of Africa model. What about fossil evidence for multi-regional? The fossil evidence for multi-regional is hard to defend for Europe and the Middle East, but it's a lot easier to fit the paleontological data from Asia. We see that there is regional similarity in the morphology of Asian Homo erectus and anatomically modern Homo sapiens. These similarities include thickened bones on the top of the cranium, a continuous brow ridge, and a rolled inferior edge of the orbit. Milfred Wolpoff, who's the foremost proponent of the multi-regional model, believes this combination of traits is unique to Asian peoples and ancient Homo erectus, and that the best explanation for these similarities is that there's genetic continuity among them. As is often the case when molecular anthropologists hear that there's a large controversy in paleoanthropology, they assume that looking at the genetic data will provide exact answers. Molecular anthropologists argue that by studying patterns of genetic variation within the human species and comparing them to predictions of population genetics, many clues about human history can be discerned. Biologists in general have used mutation rates to determine timing of divergence of various species, and they look at the genetic differences between chimps and humans as an example. They calculate how fast the mutations occur, how long it would have taken for those observed mutations to have accumulate, and they tried to use this method when examining living human populations and when modern human populations began diverging from one another. But they were unsuccessful using nuclear DNA because it mutates much too slowly for measurable accumulations to occur in 200,000 years. Many of these genetic studies, therefore, have been conducted on mitochondria, which I'll get to in a moment. But if the molecular data is correct, and if these molecular anthropologists are correct, and the replacement model is correct, we expect to see that the last common ancestor lived relatively recently in Africa. We would expect that there would be more variation in Africa than anywhere else, and that there'd be progressively less variation in younger populations further from Africa. If the multi-regional model is correct, molecular predictions would be we'd expect to see the last common ancestor lived long ago, a few million years ago in Africa, such as Homo habilis, and that there'd be similar levels of variation everywhere, as all populations have been evolving for a similar time, and there has been constant gene flow. So when they tried to eventually test these hypotheses, they attempted to use the mutation divergence method on modern human nuclear DNA to determine how long modern human populations had been diverging from one another. Unfortunately, they failed. That is, as I mentioned earlier, nuclear DNA mutates much too slowly for mutations to accumulate in the time frame that we are looking at for modern humans. Measurable accumulations of differences had not occurred. It was too short a time frame for the mutations to occur. Since then, scientists have used the same method on modern human mitochondrial DNA. So why were they using mitochondrial DNA? Well, first of all, mitochondrial DNA is only inherited from your mother. 2, and this means that the mitochondria of every living person is an exact copy, modified only by mutations, of the mitochondria of a single woman who lived sometime in the past. This woman is sometimes given the name Eve, but she's not, Eve. She's not necessarily the first woman, but rather a woman from the population who happened to survive, whose mitochondria happened to survive while the mitochondria of other women did not. Second, there's no recombination in mitochondrial DNA. So unless the mitochondria carry a novel mutation, a child has exactly the same mitochondria as their mother. And thirdly, the third reason that scientists have used mitochondrial DNA to explore questions of human variation or human evolution is that in mitochondrial DNA, mutations occur much faster than they do in nuclear DNA. So you can look at a much shorter time frame using mitochondrial DNA. A group of geneticists, Stone, King, Kahn, and Wilson, they were at Berkeley, and they looked at mitochondrial DNA variation in 147 individuals. These are people who are African, Asian, Caucasian. Of these 147 individuals, they've identified 133 different types of mitochondrial DNA, all presumed to have originated from a single type through a series of mutations. They constructed a genealogical tree of those 133 mitochondrial DNAs, and they claim that their analysis can tell us two things. One, the time of divergence of anatomically modern Homo sapiens, and the possible location of the origin of the first anatomically modern Homo sapiens. So what were the results? The most plausible route for the tree is in Africa. So the African branches all had the most mutations in mitochondrial DNA. Secondly, the common ancestor lived in Africa approximately 200,000 years ago. Once again, this is the equivalent of replacement, suggesting that the genetic data is in concurrence with the replacement model. This individual from 200,000 years ago was dubbed mitochondrial Eve. Another very interesting result of this study is that the amount of genetic variation among modern humans is really small, especially compared to other animals. And this suggests, once again, a very recent common ancestry, once again, that supports replacement. Because the more time that's evolved, the more mutations that would have accumulated. In all human populations, the genetic difference is about 0.3%. In between gorilla subspecies, the average difference is 0.6. So gorilla subspecies have twice the amount of genetic variation as all human populations. Orangutan species have 5% genetic difference, and compare that to 0.3 in human populations. Similar computations based on a single locus found that the last shared ancestor of modern humans lived between 45 to 30,000 years ago. Once again, all this genetic data is consistent with replacement and not the multi-regional model. Lastly, this study found that there's greater genetic diversity in Africa than on other continents. This could mean that on average there have been more people in Africa than on other continents, but archaeology suggests that this is not the case. Africa was very lightly populated during most of the time that anatomically modern Homo sapiens emerged. It's more likely that the greater genetic diversity in Africa represents the fact that African populations have been evolving longer than other populations, once again supporting replacement. And also, the last common ancestor for the human Y chromosomal gene also stems back to about 200,000 years ago, once again supporting replacement. What about the Neanderthals? Neanderthal mitochondrial DNA has also been explored to address these types of questions. And on average, the Neanderthals from Feldhofer Cave, they found that Neanderthal mitochondrial DNA differs by 27 base substitutions from living people, from anatomically modern Homo sapiens. Modern humans differ from each other by 8 base substitutions on average, so more than 3 times more different in Neanderthals are from modern people than humans are from other humans. So the last shared ancestor of Neanderthals and anatomically modern Homo sapiens probably lived long before the last shared ancestor of living humans, sometime around 700 to 550,000 years ago. Other mitochondrial DNA studies of Neanderthals are also consistent with these findings. Once again, the average number of differences between Neanderthals and living humans exceeds the amount found within modern humans and even between chimp subspecies. This supports the designation that of Homo neanderthalensis as an actual species versus as a subspecies of humans. It's been noted that Neanderthal mitochondrial DNA shows no greater similarity to contemporary Europeans than to other geographic regions, and that was one of the assumptions or implications of the multi-regional model. We see that there's roughly equal numbers of mitochondrial differences when compared to living Africans, Asians, and Europeans. Studies of Neanderthal nuclear DNA, there's a study between 2006 and 2010, and suggests that there may have been some admixture between modern humans outside of Africa and Neanderthals. So the Neanderthals that were in, let's say, Middle East and Europe may have, to some extent, mixed with anatomically modern Homo sapiens. A Neanderthal male and anatomically modern Homo sapien female may have been able to reproduce fertile female offspring, as with tigers. An anatomically modern Homo sapiens mother would account for the lack of Neanderthal mitochondrial DNA in anatomically modern Homo sapiens. This is obviously very controversial, Some people have suggested that the shared DNA could be to a common ancestor. Some researchers are just not convinced. It's, as I said, it's controversial. People are still talking about it. If there was admixture, it would be very slight, supporting an assimilation model, not a true multi-regional model. But either way, the idea that there may have been admixture is very intriguing. There have also been some interesting studies of nuclear gene frequency variation. These focus on polymorphic loci such as the ABO blood type, and they calculate frequency of alternative alleles with various populations, and then they construct trees of genetic similarity. These have been constructed through really elaborate mathematical analyses. The most important thing to note is that they consistently produce trees in which non-African branches join together before any of them join the African branches. So non-African populations share a more recent common ancestor with one another than they do with the Africans. And once again, this supports replacement, not multi-regional. And this is just a graphical illustration demonstrating some of these, how some of these populations are related to one another. So what's the takeaway from this? There appears to be more convincing evidence for a replacement model than the multi-regional model, but there's still debate over whether there was any admixture between anatomically modern Homo sapiens and archaic Homo as anatomically modern Homo sapiens moved outward from Africa.